Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to the Genetics Podcast. Our guest today is Frank Gleeson, who's a veteran biotech entrepreneur and operator and the co-founder and CEO of Satellos, which is a Canadian biotech that's pioneering a new approach to treating Duchenne Muscular Dystrophy. Unlike most efforts that have been focused on restoring the dystrophin gene, they actually have a novel approach that's going after a different pathway with a very different approach that I think has potential to be agnostic of the particular genetic underpinning of DMD. And we're going to go into some details about how this works and the discovery, but it's based off of, I think, a 2015 discovery from scientific co-founder Dr. Michael Rudnicki, who discovered this pathway that we're going to talk about a little bit and its role in stem cell differentiation in the muscles. And this was the birth of the company. That was many years ago. And it brings us through to today, where I think recently you've had successful phase 1b results where you're seeing some promising early impacts of the treatment. So I thank you for your work in the space so far and for joining us today to talk about everything that you all are doing.
Super. Thank you, Patrick. Nice to meet you, and I look forward to our discussion.
I would love if you could take me back to the initial founding story or kernel of the idea here. As I mentioned, this is based on, scientific finding more than 10 years ago. And I'd love to just hear how you got interested in this, how you took this from basic science through to actually, I think we could maybe form a company around this and ultimately help patients.
Sure. Love to do that. My colleague, Dr. Rudnicki, and I have known each other professionally for well over 20 years in the stem cell field. Michael is a prominent stem cell scientist. He has a past president of the Stem Cell Network of Canada, which funds research across the country in the field. I'm a past chairman of the board of that body. And that's how we first met. And we quickly discovered that we shared an interest in the idea of chemical biology applied to stem cells. So the idea of modulating stem cells inside the body for the purpose of repair and regeneration. So, of course, going back well over 20 years, at that time, we were building on genomics, proteomics. Cell therapy was becoming very prominent. The idea through iPSCs of being able to manipulate stem cells, put them back in the body in some fashion, cause treatment in that way. We were truly intellectual pioneers thinking about an entirely different approach. And I joke sometimes with not too much of a tongue-in-cheek, because this actually was part of our, martini-infused thinking. But, after the millennium, EPO became very prominent in the news because it was being used for doping. And this idea of a human protein that on its own naturally is a regenerative agent really gave us the courage to say, okay, this isn't such a crazy idea. Whether we can harness it or not, that might be crazy, but the idea is not crazy. And of course, that's what scientists need, right? They need an idea and they need a sense that it's possible. Now, fast forward a bit of time, Michael and I have created 2 companies. This is our second. The first company was focused on the idea of large molecules that might have the potential to be involved in regeneration. It turned out to be a fun experience. It got us acquired by a company in San Diego, Fate Therapeutics, many, many years ago. We gained great knowledge and experience and so on, but probably technology wasn't ready for the idea and for the idea of using large molecules. And so, we sort of swore a blood oath together that if the opportunity presented itself in a more disease-specific way, where we could think of it more from a regulatory point of view, how do we regulate the process and, find a way to drug the process, we'd be really interested in trying this again and creating a company. And of course, Michael's publication in Nature in 2015 provided the kernel of that idea, where Michael identified that a very well-known protein, a very large protein, the dystrophin protein, played a role that had not been recognized previously, which of course isn't uncommon in biology, that something might have more than one purpose. But in the case of this particular protein and this particular disease, which in this case is Duchenne muscular dystrophy, dystrophin was viewed as having only one purpose. And that purpose was essentially a structural purpose. So the dystrophin gene, which is mutated in this disease, causes either mutations in the protein or the absence of the protein. And muscle itself no longer hangs together as well as it ought to. So it destabilizes the muscle and the whole dystrophy-based complex of proteins that form what is believed to be muscle integrity and muscle strength. And we don't dispute that, by the way. We're not, radical thinkers. We're different thinkers, but we're not radical thinkers in that sense. But what Michael discovered was that the protein is also expressed in a specialized body of cells, which make up less than 1% of muscle, which are essentially satellite muscle stem cells. In the old days, they were called satellite cells, which is why we call our
It's the name.
company
Yeah.
Satellos. So once Michael discovered that, what that meant to us was that, there's something missing that needs to be there. And what's missing as a result of dystrophin being absent is a signal, a biochemical signal that the cell requires as part of its fate decision. And it is now confused in pursuing its fate, which in the case of injury, muscle injury, is to divide asymmetrically. It's to divide in a way that allows the cell to create self and non-self. So one cell that's a stem cell and one cell that's a progenitor of the end tissue it needs to repair. So in this case, that would be one muscle stem cell divides to create one muscle stem cell and one muscle progenitor cell. And when dystrophin is absent, the cascade of signaling that's required to lead to that decision, which involves geospatial reorientation of the cell, the realignment of all of the foundational machinery to create two different cell types, gets mixed up. And we reasoned that if this is a signaling issue, now we can bring into the equation the idea of signal transduction, which of course is a much more proven path. It's much more well-plowed path. And also the idea from proteomics that there are always compensating pathways.
Right.
Right. And in fact, it's one of the things that I think has bedeviled proteomics. When you look out from point A, what you see are these, maps of overlapping pathways. How do you find your way through? In our case, however, what we were saying is we know where we want to be. We know where the problem is. We can isolate. How can we find something else that might be there? Now, all easy to say, or maybe not. In retrospect, it sounds easy to say, but this took years of work, exploring this through chemical means, exploring it through genetic means, to try to identify compensating pathways that would enforce this polarity decision and would not have negative consequences. So we weren't looking for mitotic agents that stimulated mitosis. We weren't looking for any other type of proliferative agent. We didn't want to find drugs that would have toxic side effects by hitting all kinds of other proteins in the general area or downstream. We were really looking for a very specific type of signal. And we had no idea if we would find that, but that
Right.
was the beginning of our journey. Now we've identified multiple different approaches that could address this problem, this singular problem of how do you get the cell to believe the signal is there that it needs to polarize properly and then divide properly. And what over time we identified and zeroed in on was a particular protein, essentially that we kind of stole. Well, first off, Michael has a very sophisticated muscle in a dish kind of
Yeah.
screening system that we call myoregenex that allowed us to interrogate hundreds of pathways. So I'm being a little cheeky and saying we stole something. We didn't. What we were able to do once we started to zero in on pathways was steal a page out of the area of developmental biology and to really look at work that preceded us, probably by a generation, in Drosophila and other developmental organisms, where polarity is critical to the decision-making process. And in Drosophila, we were able to essentially confirm that one of the pathways that we identified, the notch pathway, plays a significant role in polarity in Drosophila. Particular protein is involved. There's a NUM-related protein that's involved. We believe that we've zeroed in on essentially the human homologue of this particular protein. We identified essentially multiple NUM-associated kinases. Not we identified them, we zeroed in on a small subfamily of NUM-associated kinases that are known in the literature. And we essentially teased them apart because there's redundancy amongst them to find the specific one, which we zeroed in on one called AC1, adaptor-associated kinase 1, which basically modulates notch signaling on one pole of the cell. Now, when polarity is the outcome you're trying to influence, creating differential expression of notch signaling between the two poles turns out to be a really clever thing to try to do. And it's effective. It changes the polarity of the cell. And in this case, function follows form. When the cell polarizes properly, it divides properly. And when it divides properly, you get a progenitor cell with each cell division. So
Interesting.
one stem cell remains to go back into the muscle and be there for the next, sort of SOS call.
Yeah.
And the other becomes a progenitor, which then takes on its own independent life and can divide 2 to the 12 times. So now you have this sort of legion of troops, if you will, that can home to the muscle damage, repair, and regenerate by actually creating new muscle cells.
So if we zoom out for a second, just to make sure I'm still tracking on this,
Yeah,
in DMD, you'll have almost like a twin set of problems. You have the
yeah,
scaffolding issue where the
yeah,
muscle is degenerating, but
yeah,
you also have this issue where the troops aren't coming in to replenish it because of this
right,
polarity issue.
yeah,
And so you don't necessarily have to fix the first issue if you can fix the second one, right? Because if you can, it's having both that's the problem. If you can boot up the regeneration engine, then if you have
yes,
some degeneration, the
yes,
hypothesis is that
yeah,
actually you can
right.
kickstart that and replace some of the loss.
Absolutely. That's absolutely how we look at this. And in that sense, we have been a little bit a voice in the wilderness for a few years because that is not how convention wanted to
Yeah.
look at this, right? So what we believe is the case now is the evidence seems to be fairly objectively clear that addressing the problem at the level of the fiber is not affecting regeneration. We believe that affecting the issue at the level of regeneration will help the fiber.
Yeah.
So ideally, one would want both. And we believe that the idea of two approaches could lead to functional cures
Yeah.
for individuals living with Duchenne. We believe that our approach, which is genetically independent of any exon or any immune issue that a child or a young person may have, has the potential to be disease modifying
Yeah.
on its own. And, I've learned from my many years in biotech that for people who live with illness, something that dramatically changes the course of their life and how they deal with that is
Yeah.
is pretty important.
Yeah,
So
and I guess there's a perfect can be the enemy of good sometimes. Like if you think
exactly.
about it from first principles, if you're able to go in and genetically edit every stem cell and fix the problem, but that requires, immunogenicity issues and solving that. Maybe could you talk a little bit about the current state of the art in DMD? Because I think it would help people level set to learn a little bit about the Sarepta
Sure.
therapy that's already on the market, some of the exon,
Sure.
exon skipping approaches more broadly, just give people a sense of what the options are today.
Yeah. So we look at it that there are, some buckets. One bucket, and perhaps today the biggest bucket, is the genetic bucket, and it has two flavors. So there's the DNA approach where we're trying to put dystrophin back into the body through a delivery vehicle. Normally AAV is the delivery vehicle. And because the size of the gene is so enormous, 4 million base pairs or some crazy number, this is not possible to package into a vector, an AAV vector. So the field is challenged to identifying a small number of repeats that can be packaged. And generally, it's about a package of about 5 repeats that go into what is called a microdystrophin package. And so now you have obviously some immediate challenges. Have you got the right ones? Do you have the right promoters? Have you optimized? And so on. These are non-trivial scientific issues. But the more fundamental problem is once you infect the muscle fiber, and a muscle fiber is usually a very elongated cell. And in fact, it's a multinucleated cell.
Yeah, I
So
remember learning
you,
about that.
yeah, so you now have infected this cell and you get an expression of dystrophin. What appears to be the case is that it does help to stabilize the fiber, but it's not clear that it's translating into significant functional benefit, which had been the expectation. Now, we believe part of the reason for that is, of course, that it's not leading to the kind of repair and regeneration of muscle that's actually required that's occurred. Now, the next flavor of genetic approaches is exon skipping. Again, you're putting something into the body, the purpose of which is to enable the expression of dystrophin in the fiber. And so, this case, the focus is on situations where a significant exon has been damaged. And essentially, the what is being delivered is a kind of pontoon bridge to cover the area of damage so that the gene can express the protein in a more complete form, not necessarily a perfectly complete form, but we're not putting the gene in, we're trying to help the body make the gene that it's not making. And this is leading now to some quite high levels of expression of dystrophin. And what has been the rule of thumb is that the more dystrophin that can be expressed, the greater the biomarker effect, the greater the
Yeah.
likely benefit. But once again, I think by objective view, that the functional benefits, at least from a statistical point of view, are modest, very modest, and not at the level that everyone had hoped. Now, maybe there'll be further improvements. I'm not disputing that. I'm not taking a shot at anyone else's work. This is not easy to do. This was what was the belief, was that the fiber was the issue. But now I think there's an increasing understanding that there's more at play. Now, there are some other approaches. There are companies focused on the heart, particularly on cell therapy towards the heart, such as Capricor, which is doing good work there and looks like has a good pathway to the FDA for approval of their approach. The heart, of course, is of course, it's the most important organ, I suppose you could say, if you want to stay alive. But this is the organ that gives out at the end of the day for patients with Duchenne. So this is something that needs to be looked at, and they're looking at that directly. There are some other approaches to address contractile damage, to try to basically limit contractile damage from movement. This is what is believed to set off all the problems, right? We move around. Human beings, we move around. We carry a load, right? We stress our muscles, and they get damaged sometimes in small ways and sometimes in large ways. But daily activity, climbing stairs, young children, climbing a tree or falling off their bicycle or whatever it is, are constantly damaging their muscles. And in the case of a child with Duchenne, they're not repairing them. So some approaches, the idea is if you could lessen the amount of contractile damage, then you're lessening the demand on the body to repair. So these are all different approaches. And I hope I've covered the waterfront reasonably well. If I've missed anyone, it's not by design or as an insult to them. But absent from this, all of these approaches is bringing back muscle itself. And that's what we're trying to do. And we believe that's complementary to virtually every other approach. If a child has received gene therapy, which with today's technology, it's a one and done experience, they would not be ruled out from our therapy at a later date. The same with exon skipping. If a child has an exon 51 mutation and has received gene or exon skipping to address that, our approach could complement that. In fact, we think quite beautifully, because now you have both an ongoing supply of dystrophin and an ongoing supply of regeneration. So it would seem in the fullness of time a nice marriage. So anyway, that's the field, and these children have a true significant challenge ahead of them in their lives, and they also, in my experience, any that I've met, they have the most remarkable spirit. So I cheer for all of the other approaches because the kids deserve it. They deserve help. And in our case, I was at Cure Duchenne last week, and I presented at their annual meeting, and I made the joke on the stage that as a Canadian, I'm obsessed with hockey, ice hockey. And, when you play hockey, you learn that an assist is very valuable, that making the assist is just as important as scoring the goal. And that's how the team gets built. And that's what we are thinking about in this disease state. The assist we want to give is an assist to the body to heal itself. If we can assist the body in restoring the normal process that it is trying to establish, the bodies of these children have not given up. They are trying to repair muscle, but they don't have all the tools to do it. So we want to give them an assist to do that.
I love that. You're very good at metaphors, by the way. Has anybody ever told you that?
Thanks.
We got the hockey assist. We got the pontoon boat. You're very good at connecting those dots.
Right.
On that same topic, though, so you presented your phase 1A data pretty recently, which showed the compound was safe, well tolerated. I think hitting the pharmacokinetic profile that you're looking for. You're moving towards phase 2. You're in 1B right now, where you'll start to see some signal of how this might be working in patients. I would love to just hear a little bit about the clinical development plan more broadly. What are you expecting to see, and any high level timelines that you're thinking about for this?
Sure. So we've approached the development, of course, to be consistent and follow the regulatory rules and regulations. But we're very mindful of what has gone before and the fact that we're dealing with a population of individuals who are on many different medications from a very young age, some of which, are actually quite difficult on their systems and on their bodies. So we've wanted from the beginning to try and design a molecule that was highly tolerable and, as best we could, had a minimal side effect profile. So, in addition to adding to the novelty of what we're trying to do, this has guided our thinking. And so we've approached this that we've taken our time in preclinical development. We've tried to design the molecule. We've tried to move the atoms around in a certain, to meet certain objectives in the animals as we're, doing our preclinical work. The PK is very important to us. The profile, the dwell time of the drug is very important to us, all because we want clearance as quickly as possible. We don't want to interfere with the mechanism we're trying to manipulate, and we want to, minimize the possibility for side effects. So specificity to this target was extremely important. We spent a long time working to get a highly specific inhibitor of AC1, and we believe we've achieved that goal. And we, designed a safety trial, a phase 1 safety trial in healthy volunteers of 72 individuals. So we did 9 groups of 8, split between single ascending dose, multiple ascending dose. Of course, we looked at safety, we looked at tolerability, and it's very clean. And certainly in the dose range that we expect to be the clinical dose range, it's remarkably clean compared to many drugs I've worked on in my life or seen. So that's very encouraging to us, and we're pleased to see that. And the drug has the PK properties that we were looking for. It meets the time above the Cmax. For those of your listeners that understand what we're doing, this is a Cmax driven mechanism. We've got the PK, we've got the time above Cmax that we're looking for. So all of those variables we wanted to build in, and we saw we did that in the phase 1A. That was important to us. And then the next piece in this, again, recognizing in a way the special nature of this population of individuals, we wanted to ensure that the PK, when we went into the target population of children, as best we could, that our PK wouldn't be affected by standard of care, which is corticosteroid treatment.
Right.
And I'm not criticizing, by saying that, I'm not criticizing corticosteroid treatment. I'm only stating that we recognize all of the children that we'll be treating,
Yeah.
not all, but the
Vast majority.
will be on steroids of one form or another. And it's a very difficult experiment to do in the animal models because the steroids overwhelm the animals.
Right.
So it's hard to tell whether the PK of your drug is being affected by the steroids because the dose you're driving starts to harm the animals. So we felt that if we could get into if we could work with a population of DMD patients, we could answer this question. And so we decided to approach, we were doing our studies in Australia, so we decided to approach MDA Australia and ask them, what they thought of the idea of us doing a small study in adults with DMD, because we had the tox profile that we could do that. And we had coverage for 28 days from our initial tox studies. So we could treat for 28 days. And, the adults are generally on steroids and have been their whole life. So we did a small study of up to 10 patients with regulatory approval, of course. And we looked at safety in that population, and we looked at PK in that population. And we also did a number of exploratory measurements, not really knowing in any way what we might see, because very, very little, frankly, from even a medical point of view, but certainly from a biological point of view, very, very little is known of the composition of muscle in adults with DMD. We know that it breaks down consistently and progressively and ultimately begins disappearing, but you just don't really know because the data isn't there. So we measured things that we thought we could measure without putting too much of a burden on these individuals who sometimes have to travel great distances to come to a clinic, and it's very arduous for them. So we looked at grip strength, we looked at pinch strength, we tried an experimental measure of upper body sort of movement, because of course they're stationary in a wheelchair. We looked at forced vital capacity, and we took blood samples to look at serum. And even something like that is very difficult in some of these patients because, it's hard to even get a vein.
Yeah.
So we compiled all of that information over a 28-day period. We were able to recruit 5 individuals who all completed the study. Had we been able to recruit 10, we would have, but many patients were even too sick to be in a study with a small molecule. So we decided once we had 5 that we would stop there and complete the study and see what we saw. And so in that body of patients, again, almost nothing on the side effect profile, very, very clean. Drug-related effects, of course. These patients sometimes had their own challenges because of the nature of the disease they live with. The PK was not affected by the steroids, which was really, really warming to us because it means all of our planning and modeling for dose should now translate quite nicely going forward. And we saw, for the most part, across the measurements, with one exception, we saw essentially stabilization. So, on things like pinch strength and forced vital, essentially we saw, maybe a 10% change, sometimes positive, but basically either stable or a small positive change. We felt this was within the margin of error. When we talked to professionals, they basically said, a 10% change, even in this population, you can't really say that's drug related. So we've basically said to the world, stable on those measurements. But what we did see in grip strength across the 5 patients was that 3 patients showed significant changes, well above 10%, in both left and right hand. And overall, for those 5 patients, now you statistically do all 5, a doubling of grip strength. So that's in 28 days, a doubling of grip strength, which numerically was added 2 kilograms of strength. Now, we get asked the question, well, okay, so what can they do now they couldn't do before?
Yeah, that's exactly what I was going to ask.
So, we look at it that in the way, what we've seen in every other study we've ever done, all the preclinical modeling in mice or dogs, is that force is what we see first. Increasing force is what we see first. And it's a leading indicator, not a trailing indicator. That's what we've seen. And what it indicates is that muscle cells are being made. That's what it indicates. And so if that's holding here, then actually what's happening biologically is new muscle is occurring. In terms of what does an absolute improvement of 2 kilograms mean, at this moment, the truthful answer is nobody really knows. But what patients tell us is that when they are older, their hands are, frankly, what they have left. And it's their conduit to the outside world because they need that for communication purposes, online, and so on. And so anything that helps them maintain strength or increase strength is important. And what I've experienced in meeting patients of all ages, but particularly meeting adult DMD patients, is that it can be extraordinarily difficult for them to shake hands.
Right.
So if you extend your hand, which often I feel all individuals still want human contact and touch, they will try very, very hard to extend their hand. So would a couple of kilograms of extra strength allow them to do that? I think it probably would be helpful in that regard.
Yeah.
Would it allow them, to bounce a basketball? I don't think so, but it could make a big difference. So we look at it as a relative situation, relative to the individual. So a very young person, we would reason that the impact of regeneration could be dramatic. A much older person, in an absolute sense, it would be modest.
Yeah.
But from a living life perspective, that could be just as
Yeah.
important.
May still be very impactful. As you expand out to the phase 2, do you have a sense of how many patients you need to enroll to see some of these effects? And also, are you able to push the 28-day window
Yes.
out at all?
Yeah,
'Cause,
yeah.
there's a major
Yeah,
compounding here.
yeah. So a couple of things. Yes, we debate this all the time. How many patients would be enough? We've seen many, many studies where, people are reporting results on a small number, very small number of patients. We're trying to show function. We don't want to misfire because we didn't enroll enough patients.
Yeah.
And, we don't want to make it such a big study we can't actually conduct the study. So we've run our statistics. We've looked at, projecting from the kind of changes we see in our models and so on to try and get an N size for a study. Our next study will be a phase 2 study in children. At the moment, the only guidance we're giving to the market until we finalize the protocol is that we'll be working in children under the age of 12, so still ambulatory children. We'll be looking to do 51 children, and 51 because that's divisible by 3. I remember from my grade school math, they have to add to 6 to be divisible by 3 or something like that, whatever it was that had, whatever it was, it has to
Any
divide by 3.
multiple of 3.
Yeah, multiple of
Yeah.
3. 5 and 1 divides by 3. So good. And we'll do 3 dose groups. Placebo will be one of them. It'll be a placebo-controlled study, a dose group A, and then dose group B, which will be a higher dose than A. And we're working those details out. We expect to be filing with regulatory bodies across the world in the July timeframe, possibly July, August, but sometime in that portion of the third quarter, so that we can, hopefully be in position that we have approval to move forward before the end of Q3, at least in some jurisdictions. This will be.
Congratulations. It's
Thank
exciting.
you. Thank you. Oh, we're really
Yeah.
excited. And it will be a 3-month treatment period. So the study for each individual child will be 3 months. Generally, that means about a 4-month study because a couple of weeks beforehand prep and a couple of weeks of follow-up comprises the sort of time frame in the study. And as I said, we hope to be, dosing patients certainly before the end of this year and hopefully very, very much towards the end of Q3 or early Q4.
Yeah.
And then in parallel to that, a kind of agreement that we made, I suppose you could say, with the volunteers who participated in the adult study was that we would ask for approval to run a follow-up study from the Australian regulators. So we will be running an 11-month follow-up study. So 11, the first month makes 12. And they've been very, very accommodating, I guess you could say, the regulators to allow us to start this study and then on a rolling basis submit our toxicity kind of data for longer and longer periods of treatment. So that allows us to get going and not have to wait until we've done a 9-month or a 12-month study and tox study before we can start this program. So we hope to also be generating data on a regular basis in that follow-up study, and we'll invite the participants back for that. And, we're talking to the regulators now about whether we could bring additional patients who may now want to try again to be in a study because they know it will be longer
Yep.
maybe they'll want to try again. So we're doing whatever we can to work with the communities and also do whatever we can to get data that will persuade us and persuade others that there is a potential for benefit.
Yeah. You raised $55 million in 2023, which is it's been very hard for biotechs in general for a very long. And I know prior to Satellos, you spent a lot of time in venture capital. I'd love to get your perspective on what it takes for biotechs to cross that funding bar in this kind of climate, because it is far harder than it was in 2021, 2020 by any stretch of the imagination.
Yeah. And we raised money again at the end of 2024.
Right. I missed that. Congratulations.
Just same amount just before Christmas on the 24th of December, which was the Friday, before everything shut down. So we really feel fortunate that we closed that deal before the new year.
Yeah, I'm seeing that here. So 40 million in 2024, right?
So in both cases, it was 40 million US, which is 55 Canadian. So to your question, the most important thing is persistence. You just cannot take no for an answer, or you cannot be deterred by someone saying no. You have to keep at it. You have to keep going out there. You have to keep refreshing and taking your story to people. That's number one. Number two is, well, it's probably self-evident. I don't mean to be trite. You really do have to have something of interest. So what is it that one has that's of interest? And to whom is it of interest? And sometimes that's hard to tease out. For us, early on, what we had of interest was this fascinating science. I mean, my colleague Michael and we were just like beside ourselves with this discovery. But I can tell you, a large part of the world really didn't care.
Not everybody else was, but you only need to find one.
You only need to find one. So that's what we just, had to focus on.
Yeah.
But as we moved along and as we were able to develop more data and we were able to answer more questions, then the pool of interested parties expanded quite significantly. It still didn't mean they would all say yes and give us money. What it meant is they might follow us, they might encourage us, they might be honest with us and say, never in a month of Sundays will we be interested in this because it's not our thing. And that allows you to get focused, right? That allows you to zero in. So I think it's persistence. I think it's a case of understanding what you have, why it's important, to whom it's important, and then, kind of packaging it. And I don't mean that in a pejorative way, but no matter how smart people are, no matter how many degrees they have, when they're meeting with you, they may have 15 minutes. They may have 30 minutes, but they don't have hours and hours and hours to dig in and figure it out. And it's the job of the presenter to make that amount of time matter and not to get mad when somebody is not understanding or is preoccupied, because who knows, they might have lost an investment earlier in the day, or they might be working on something else, or they had a fight with their family. Who the heck knows? It's all about people. So I just think it's very much that, and it's not easy to figure out what it is you have and to whom you should be selling it and why they should care. But if every time, and we would do this every meeting, literally, we would go through this. Okay, so we're talking to this group. What are we going to talk about? Why are they going to care about this? And, you want to get to those punchlines as fast as possible. And what I have found is, as long as you can back it up with the due diligence, you can make a lot of statements. You will get the audience, particularly the US venture world. They're very sophisticated. They will accept your presentation, which is not to be misconstrued with getting suckered. They will accept. They will listen. They will accept. If they like it and they want to dig in, you better back it up.
Yeah.
That's where deals fall apart.
People would not realize this listening to you, but you didn't start off in science. You spent most of your career in business, but it seems like you found a passion and a love for science that is clearly, transcended or accompanied that love for business. And I can't help but wonder if that the combination is really powerful here. I, I think sometimes scientists can fall in love with the science to the detriment of understanding how do we actually commercialize this and how do we make it sustainable. And it seems like, if you can balance those two in the right way, that's where the magic happens. Do you see it that way?
Yeah, I do. And I've seen, both sides of that coin. The scientists that they are in love with the science and they lose the ability to be objective. And I've seen the business people that can't cross over far enough to actually understand what might be brilliant about the science.
Yeah.
And I think that's the secret is I like scientists. I mean, I like sitting down and talking to them. I like learning. Not that other people don't, but I genuinely like that. But I don't want to do their job. I do not want to be at a lab bench. I do not want a pipette in my hands. I would be an utter disaster. I can barely boil an egg. But just would be a nightmare. And I know that. So I live it vicariously. And where I've been most successful working with scientists is when they feel the same way about business. They're smart enough to understand it. They're curious enough to want to know what's going on. And then they just say, thank God I don't have to do it.
Right.
That works really, really well. And to me, that's the secret is mutual respect, alignment around what you're trying to achieve, and a joint passion for whatever the topic is, right? I mean, this is my thing. Someone else might be in another area, but if they're all passionate about it, it's the same thing. You get juiced up. You don't have to pump yourself up to go into a meeting.
Yeah. I think that's a brilliant note to end on. This has been hugely educational for me, learning about the space, learning about the unique pathway you're going after. And it's caused me to think about this disease in a different way, but also think about how you can approach in a more agnostic way some of these diseases and break across some of those silos. So thank you for this. I really appreciate you taking the time.
Super. Well, thank you. I've enjoyed the conversation. Thanks for the questions, and look forward to whatever's next.
Absolutely. And we'll be keeping our fingers crossed. Probably some of the most tense moments for you is waiting for the data lock to go in and the analysis.
Every time data's going to be coming out, we all have a panic attack.
Trouble sleeping. Yeah. Well, yeah, thank you again for spending the time with us.
Okay. Thanks a lot. Bye now.
Thanks everyone for joining us, and we'll see you next time. That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible, in particular Amy Cousins and Sonia Shah who produced the show, and James Pierce from Selective Frequencies who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.